welcome all participants on the first Fermentis wine webinar event, Central Europe and let's say Caucasus and Central Asian region. My name is Yannis Topalidis. I am a regional sales manager of Fermentis, responsible for Fermentis activities in Central Europe. Let me introduce all other participants and mainly moderators and presenters. So first of all, thanks for Victoria Troianu. Victoria is manager of Innovino. She is a chemist and enologist. She is a Fermentis distributor in Greece. Thank you for participation on this event. And she will give us also a small presentation about implantation tests we did with SafOeno SH12. Also, thanks Etienne Dorignac accepted our invitation. Hi, Etienne. Etienne is the Fermentis technical manager, chief enologist of Fermentis and chemist. He will lead the presentations today. And also, thank you, Gilles Gueumar. Ciao, Gilles. He is a technical manager, technical sales support manager. He is technical people person, and he gives support to the sales people for us. Gilles is bioengineering, specialized bioengineer, and specialized on brewing science. It's not a problem. Gilles, oh, we like brewers as well. Participants came, as I saw the list, from all of Central European countries, mainly from Greece. Thanks for Vicky, initiated this webinar. And most of participants are from Greece, but I saw also participants from other historical wine regions like Armenia and Georgia, and also from Romania, from other Central European countries, from Poland as well, and from Hungary as well. Now we introduced all of us, and now I ask Etienne to go to the first slide, because I would like also to introduce in 2 minutes Le Saf Group, our mother company, and then Fermentis in 2 words. So Etienne, please, you are the lead, you are the...
Yeah, normally you see the screen already.
Pardon?
Normally you have the screen now.
Yeah,
we have the screen.
Thank you, Etienne. And
hi, again.
Hi again.
Okay, thanks back, Vicky. Thanks, you get back. I introduced all of you for all other participants, so that's it. And now go on, Etienne. Please go to the next slide. Thank you.
So that's the agenda. You see the agenda?
Yes, this is the agenda. Etienne, please go through on the agenda.
Okay, so on the agenda there will be a bit of introduction by Yannis about Le Saf and Fermentis, and then we will have actually 5 parts. We will have a global overview of the impact of yeast on one aromatic profile and a focus on thiols, to know how to release the thiol. Then secondly, we'll have the process, what we put in place as a selection process of the SafOeno SH12. As far as we selected the SafOeno SH12, we produced it as a dry yeast. So Yannis will explain a little bit the easy-to-use production process and the result on the dry yeast. Then I will move on some guidelines to optimize thiol expression. So to use the SafOeno SH12 as it's best to express the thiols. And then we will finish by the SafOeno SH12 field trials. So a global overview of what we did, and also a testimony from Vicky about, let's say, her experiment in Greece with this yeast. And then as a conclusion, we will have the characteristics of the SH12 and a Q&A session. So if you have some questions, actually you can type them on the chat, and maybe we will review them at the end, let's say, of the presentation. And Gilles will pick up some actually interesting questions, and we will be able to reply to that at the end of the session, if it's okay for everybody. So I let the floor to Yannick for the Le Saf description.
Okay. 2 minutes about introduction of the SAF group. Thanks. So the group has. Did you hear well me? Because I would like to ask Etienne. I have some echo also on your voice.
Yeah,
so
maybe Vicky, you have maybe the microphones, but I will shut down my microphones.
Okay.
Yeah, you can go.
Okay, great. So the company, Le Saf Group, has established more than 160 years ago. So the base is in Lille. Today, the company has a 2 billion euro turnover. We work more than 10,000 employees, 70 nationalities. So the company is quite a multinational one. We have production or operational companies in 50 countries, 66 operational companies we have up to now. And our products, we reached 185 countries. We can say very briefly that each third bread made with Le Saf yeast in the globe. R&D, we have 10 R&D centers and close to 600 experts, biologists, biochemists, engineers for applications, also enologists, brewing science people. So we have quite range of experts. So next one, Etienne. If we see the structure of the activities of Le Saf Group, we can say that we have 4 main segments, 4 main parts of our activities. First one is the baking part, the main part of Le Saf activities. We produce products, different kind of yeast and yeast derivatives for baking industry. The second one is healthcare. It's mainly focused on plant protection, animal feeding, and pharmaceutical industry, products for pharmaceutical industry. The third one is, we call it industrial biotechnology. It's mainly products for bioethanol production and other green energy sources, so nutrition and also yeast for high gravity fermentation and for special conditions. And what is most important for us today is the food, taste and pleasure part, where you can see the products we sell for and we produce and developed for food industry. And inside in this segment, you can find Fermentis business unit, which is dedicated for beverage fermentation. And if we go a little bit deeper, but not long, we can say that Fermentis is a Le Saf business unit in charge of development, marketing and sales of innovative, qualitative products, services for beer, wine, spirits, and all other potable alcohols. If you see the wide range of alcohols we can find in Japan, sake, or in Greece some other alcohols like hopefully tomorrow Retsina, and we have also other tequila for Mexico. So all other potable alcohols and spirits, we also work with this. So we say that our motto is Fermentis is the obvious choice for beverage fermentation. Thank you very much. No more time for introduction. I give back the voice for Etienne. So Etienne, please take the lead for the next.
Yeah, thank you, Yanit. So we will start this webinar by the global overview of the impact of yeast on wine aromatic profile. So actually, some of you already saw this slide. I think it's just a global overview of the main pathways that you have through the yeast to produce aromatic compounds. So for sure, the first way you use the yeast is to ferment. So ferment the sugar and to convert into ethanol. That's the glycolysis pathway. Through this glycolysis pathway, actually, you already have some interesting compounds that are produced by the yeast, like acetic acid, acetaldehyde, that are not really, let's say, compounds that you look like for wine application because they are associated with all flavors. But you have some compounds like glycerol that can divert the carbon source to glycerol production that can be useful for and beneficial for the wine process. But one of the main compounds that you produce through this glycolysis pathway afterwards is the acetyl-CoA. This acetyl-CoA compound is actually involved in the production of fatty acids. These fatty acids, actually, when they are released as fatty acids outside the cells, they have some cheesy nose. It's not really interesting. But what will be interesting is that the fatty acid acetyl-CoA that will be involved in other aromatic compounds. The yeast can use the glucose fructose, but the yeast can use as well nitrogen sources. And these nitrogen sources, you all know that that are amino acids or ammonium. These actually can enter the cell actively and then be converted for sure to the protein synthesis, but can also be integrated in the production of raw materials like higher alcohols through the Ehrlich pathway. And these higher alcohols have already an impact, an aromatic impact, and will be also involved in the production of other aromatic compounds. One other source actually of nutrient that can enter the yeast cell are sulfates. And the sulfates enter the yeast cell and the sulfites as well, and go through the sulfate reduction pathway to produce what we call the sulfides. And the main sulfide we know is the H2S. For sure, if we accumulate the H2S, and if it's the H2S is actually released outside the cell, it's really these reductive nodes that we don't want. But the thing is that the yeast needs to produce this sulfide. Why? Because this sulfide will be associated with amino acids to produce sulfur amino acids, and these sulfur amino acids are essential to the function of the yeast. What is interesting as well is that actually one of these sulfur amino acids that is called cysteine is actually also the base of the production of glutathione. And glutathione, you know that in the wine, and not only in the wine actually, but in all segments, is really interesting compounds in the yeast, produced by the yeast, because it's a very strong antioxidant. For sure, if you produce sulfur amino acids like methionine, and these methionine go through the Ehrlich pathway, the problem is that you can also produce some higher alcohols like methanol that has some bad aromatic features like cabbage or these kind of things. Among the aromatic, let's say, compounds that the yeast can produce, I tell you that the higher alcohols are very important, and the acetyl-CoA as well, because if they combine each other, it's an esterification, they do what we call the acetate esters. And these acetate esters are really impactful in terms of flavors when you use specific yeast. So, for example, when you start to take phenylalanine as amino acids or leucine as amino acids, this phenylalanine is converted into 2-phenylethanol, the rose flavor, as a higher alcohol. And this higher alcohol actually can be combined with the acetyl part to produce the 2-phenylethyl acetate that has also a rose flavor, and that is very impactful. If you do the same with leucine, you produce isoamyl alcohol, and this isoamyl alcohol is esterified into isoamyl acetate with the strong banana candy flavors, as everybody knows. Then the second part actually of the esters can be as well the acetyl combined with the ethanol. In that case, it's not really interesting because it's producing ethyl acetate. So ethyl acetate as a certain level is okay, because as a certain level or threshold can help exhausting some flavors. But if it's too high, is the thing that you all know is the nail polish, these kind of aromatic compounds that you don't want to have in your wine. Another type actually of esters that is very, very interesting for the wine aromatic profile is actually the fatty acid pathway. The fatty acid pathway, the fatty acid can link to ethanol to produce what we call the ethyl esters. And these ethyl esters are very interesting. So it's like ethyl decanoate, ethyl hexanoate, octanoate, because these ethyl esters have distinct flavors, very fine flavors, about like a fruit cobble. It's all other fruits than actually banana or candy flavors. For sure, these ethyl esters can be branched or linear, but they are all coming from fatty acids and combination with ethanol. So all these compounds, all these aromatic compounds are produced through the yeast during the fermentation. And that's why actually we call them fermentative aromas. So whatever the matrix, whatever the grape varieties, whatever the winemaking process, you will produce during the fermentation these kind of aromas, and this certain level of aromas will depend on the yeast you choose. Another type of aromas are what we call the varietal aromas, and this is what we will focus on today. Is actually a specific varietal aromas that is called the thiols. These thiols actually are depending and really related to certain grape varieties like Sauvignon Blanc, Colombard, Syrah, or this kind of grape variety, because they have thiol precursors, what we call thiol precursors, that are precursors, aromatic precursors that are not volatile and not aromatic, and needs to be taken up by the yeast and then cleaved into the volatile and the corresponding volatile thiols. So for example, the cis-4-MMP, so the 4-MMP linked to cysteine will be taken up by the yeast, and the yeast will be able to cleave this cysteine 4-MMP to release the 4-MMP, and this 4-MMP is the volatile thiols that produce blackcurrant flavors or box 3, boxwood flavors. The same with 3-MH. A particularity of the thiols is that when you look at the second thiol, that is the 3-mercaptohexanol, hexanol is an alcohol, is like a higher alcohol. And as you saw in the bottom, the higher alcohols can actually link to the acetyl to produce acetate esters. So it's the same with the 3-mercaptohexanol, actually. It will be able to link to the acetyl-CoA and to produce the corresponding ester, that is 3-MH acetate or 3-mercaptohexanol acetate. And this acetate has these distinct passion fruit flavors that are really interesting, for example, in the Sauvignon Blanc. So thiols, it's interesting. Thiols is linked to varietal aromas, but also to the fermentation that will produce these esters that are thiol-related. Another type of varietal aromas is also what we call the terpenes that you have in the muscat or Gewürztraminer, Riesling, Viognier, these kind of actually compounds. This is the same. The only thing is that the precursor is actually linked to sugars, and the yeast will cleave this sugar to release the terpenes or the terpenols that are the aromatic flavors, mainly the citrus and floral aromas distinct from muscat or Gewürztraminer. So this is a global overview. For sure, at the end, it's a bit complicated to see all these pathways. But actually, that's the overviews of what the yeast can do, either through fermentation only, whatever the matrix, or sometimes depending on the matrix. So if you have precursors, aroma precursors, what the yeast can do to release from these precursors these interesting aromas. So if, as the SH12 was focused on thiol release, we will just focus a little bit on thiol release and specificities. The main thiol, as we saw, are 4-MMP, 3-MH, so with box tree, blackcurrant, or grapefruit, passion fruit aromas. And as we saw, actually, these are released from grapes precursors, depending on the yeast you choose. And this 3-mercaptohexanol will be converted during the fermentation into its acetate esters, and this acetate esters has the passion fruit box tree aromas with a very, very low perception threshold compared to the others. So if we enlarge a little bit the pathway, you still have this pathway to release, let's say, the thiols. But if you see, actually, these pathways, you have a lot of genes involved in this pathway. So the genetic of the yeast will be crucial for its ability to release correctly the thiols. So for example, the main genes involved are, the first ones are OPT1 or GAP1, and these genes are actually related to the protein, the permease, that will actually allow the yeast to take the precursors into the cell. So as far as you will have a control on these genes or an activity on these genes, and so these proteins, permease, you will control a little bit the uptake of the thiol precursors into the cell. So it's a critical point. Then you have the IRC7 and STR3 that are the main genes, let's say two of the main genes that are involved in the beta-lyase activity of the yeast. This beta-lyase activity of the yeast is actually the activity that allows the yeast to cleave or to degrade, let's say, the precursors, cysteine-related precursors or glutathione-related precursors, into the volatile thiols. So from cis 4-MMP, for example, to 4-MMP, or cis 3-MH to 3-MH. So these genes actually will be involved not in the uptake, but will be involved in the release of the thiols. And then, as we saw, you have a specific thiol that is the 3-MH acetate, and this one is actually the esterification of 3-MH and acetyl-CoA. And one of the main genes actually involved in that is ATF1, is an acetyltransferase. So transfer of an acetyl to the 3-MH to do this 3-MH acetate. So this gene mainly will be involved in the conversion of 3-MH to 3-MH acetate. So if you do this thiol focus, for sure the conversion efficiency will depend a lot on the strain you use, because some strains will have more activities here or more activities here, or more activities here and will release different concentrations of 4-MMP, 3-MH, or converted more or less 3-MH into 3-MH acetate. So I just want to focus now on the IRC7 gene, for example, because it's very easy to, let's say, to know if the IRC7 gene is active or not in a yeast. It's a genetic experiment. So about the IRC7 gene specificity, this is the gene actually in the genome, and this gene can appear in a full-length version. Okay, as far as you have the full-length version of this gene at the DNA level, this gene will be transcripted into RNA, but this RNA will be complete because you have the full version of the gene. It will be complete. And this RNA will be then translated to a protein. And this protein is, as we saw, is the beta allele. As far as you have the full-length version of this IRC7, you have the full RNA, and so you have the full protein. And this protein is active. So if you have precursors, these precursors will go into the protein and cleave the, let's say, will be cleaved, and the 4-MMP will be released. The problem is that in most of the wine yeast, you have a short version of the IRC7. So you see here, you miss the 38 base pair. It's called the short length version. If you have this short IRC7, for sure, when you transcript, you will have a short RNA. And when you will translate, you will have a protein that is not complete or not having the good conformation, meaning that the beta allele activity of this protein will be nothing. So if you have the same precursor, it will not be able to go and be cleaved. So meaning that you will not have the possibility to release, for example, the 4-MMP from the precursor. So what is interesting in the selection of yeast towards this gene is, as I told you, most of the wine yeast that you can isolate in the vineyard have 2 copies of this gene, but these 2 copies are the short version. So with a deteriorated beta allele. Meaning that it will be interesting in the selection process to be able to select yeast if you want a thiol releaser, to select yeast that will be able to have this full-length IRC7 version. And if they have this full length and 2 copies of this full length, you will favor actually the activity of the beta allele, and so the release particularly of the 4-MMP. So with this said, how we selected and evaluate the SafOeno SH12, our new yeast? First, through a genetic and phenotypic screening. So this genetic phenotypic screening was done it's quite simple, but it's interesting to do that. We saw 3 kind of things. We assessed the killer activity, the sensitivity of the killer activity of the strain. We assessed the length, so determine the length of the IRC7 gene for sure. But third, we also let's say look back to the hydrogen sulfide production, because most of the yeast actually that are selected as a thiol releaser can have some problems of reduction or excess production of sulfide or sulfite. So it was really interesting to look at these 3 first feature. And actually, we did that in partnership with the IFV, so Institut Français de la Vigne et du Vin, because we did a partnership and we screened their own yeast collection towards our features, these features. So to give you an idea, from 300 strains, we established the killer status, killer, neutral, sensitive. Then we took the killer strains. We assessed the H2S score, so the production of H2S. And then at the end, we selected the H2S1, so the very low sulfide producer, and we assessed the IRC7 genotype, either IRC7 long, or long, short, or short, short. And we did actually the same with the H2S score 2, so not very low producer of sulfide, but low producer of sulfide, so still acceptable. And we did the same, so we looked back on the IRC7 long, or long, short, or short, short. And so from this kind of first screening, first selection, we selected strains, first that were killer, with low H2S production, and with the IRC7 long actually version. Then for sure we did a lot of aromatic and sensorial screenings. I will only take one of them just to let you know what was the results and why we selected the SafOeno SH12. So for example, this is a microvinifications. For sure we did several ones. It's just an example. So here is on a Sauvignon Blanc, still with our partner IFV. And actually, what we did, we tested 3 strains, SH12 and the reference 2. Reference 2 was actually IRC7 long. So both of them were IRC7 long. And the reference 1 was IRC7 short. We ferment, let's say, at a temperature, normal temperature for thiol release. And what we got. So that are the results, or let's say, of the analytical parameters or the duration of the alcoholic fermentation. So what you see is that compared to the commercial reference that are referent in the market, the SH12 was regular fast kinetics, so no problem of kinetics compared to the reference. This yeast was keeping actually the acidity, so not degrading the malic acid and keeping a good total acidity. In terms of SO2, it was really interesting because compared to some references like reference 1, this yeast was selected to produce not a lot of sulfites. For sure, it's a medium producer of sulfites, but it's acceptable and not too high. And we selected as well, for sure, a yeast that were in the average on the VA, on the volatile acidity production. So that's the analytical parameters and of alcoholic fermentation. But the most interesting for us, as far as we have a clean fermenter and fast fermenter, it's to see the aromatic parameters at the end of the alcoholic fermentation. So here, I don't know if you're used to these kind of things, but it's a representation of all the compounds, all the aromatic compounds produced by the yeast. And that's a PCA, so it's a principal component analysis. So it maps actually the yeast here that are the variables. Actually, the yeast are mapped according to the aromatic compounds they are producing the most. So if you see, among all the strains we, let's say, we evaluated, you have the reference 1 here, the reference 2 here, and the SH12 here. And what you saw that this part of the graph corresponds to 4 MMP high concentration and 3 MH high concentration. But not only that, it's also ethyl hexanoate, ethyl hectanoate, ethyl butanoate, ethyl decanoate. So meaning that this part of the graph is the release of thiols, 4 MMP, 3 MH, but it's also the production of ethyl esters, and this is related to these 3 strains, among whose you have the SH12. The reference 2 is actually here in between. So we'll produce a little bit of 3MH, 4-MMP, we'll produce a little bit of acetate esters as well. The reference one, if you remember well, the reference one is actually IRC7 short, so meaning that has a disability to release correctly the thiols. And what we saw is interesting, because this reference one is in this part of the graph, so meaning a lot of acetates and higher alcohol production during the fermentation, but as well the conversion of 3MH in acetate of 3MH. So meaning that this yeast strain will release a small amount of 3MH, maybe no 4-MMP, but this 3MH will be converted very efficiently to acetate of 3MH, because this yeast has the ability to produce a lot of acetate esters. So it's very interesting to see in only two graphs here, the main difference between the yeast you can have: high producer of acetate esters, high producer of ethyl esters, ability to convert the higher alcohol into their acetate, and ability to release the thiols from their precursors inside the grapes. So SH12 is definitely a yeast that is able to release quite a lot of 4-MMP, 3MH compared to the others, especially 4-MMP, and produce during the fermentation mainly ethyl esters and less acetate esters. If you compare these to the sensorial analysis, what is interesting is to see that the SH12, if you compare to the reference 2 or reference 1, you will have actually more aromatic intensity and complexity, because ethyl esters are bringing complexity, more thiol types of aromas, and in that case, so going along the fruitiness. In terms of tasting, for sure, we keep the acidity, so that's interesting. This acidity can help in this type of wines, so young Sauvignon Blanc, to have also a high aromatic intensity in the mouth and persistence. So that's an example, let's say, showing part of the selection and part of the evaluation, but why we actually made this this selection of SH12. So I will leave the speech to our friend Yannick, because this yeast, we selected it, but we had to produce it under a dry form. And you all know that we have a specific process to produce our yeast under a dry form that is called the easy-to-use process. And I will leave Yannick the speech to talk briefly a bit about that.
Thank you, Etienne. Thank you your nice presentation up to now. So you can drink a little bit your coffee. Easy to use is an application brand. Basically, it's a certification on our yeast that allows the winemakers to inoculate the yeast in a flexible ways. So what does it mean? It means that our products that label with easy to use label or have this certification, then you can directly pitch on the mast, either directly pitch on the mast, or you can rehydrate it in a different extreme conditions, keeping the high and preserved viability in all conditions of the yeast, also maintain the fermentation performance, and also finally no difference or equivalent organoleptic profile you can get by the end of the fermentation, whatever is the mode of preparation. So let's see some figures about this. Please go again. Thank you. basically easy to use is when you ask what is easy to use and why our yeast are easy to use. So why are so robust? Why we can have this? The answer is that we have the know-how, as I told you, how to grow the yeast, how to create, how to produce the biomass, and then we know we have the know-how how to make it dry, how we can preserve and keep the integrity of the yeast membrane in order to be, let's say, viable after rehydration and during before the fermentation or during the inoculation. So now we can see what happens, what is the influence of rehydration conditions on the viability of the yeast during in different conditions of rehydratations. So what you can see, unfortunately my mouse, you cannot see it, but on different conditions are temperature, medium, different kind of medium, and stirring conditions. On the horizontal axis, you can see the temperatures. We do it 12, the rehydration, we do it 12, 17, 27, 37 Celsius. Also the agitation, the stirring conditions was without agitation, moderate agitation, and volatile agitation. And the media, also we applied 3 different kind of media, water, and different concentration of sugar source media, 10% fructose glucose 5,5, and 30% fructose glucose 15% in ratios. So the different medium, different temperature, and different stirring conditions, especially quite extreme conditions, you can see that on the viability of the yeast after rehydration are the same. The yeast keeps its viability close to 85–90%, which shows us that our cells has a high robustness thanks for the our know-how and thanks to the production, how we produce this yeast. We did at the moment, we have at least all our yeast are easy to use except of 3 of them. So UCLM 325, 377, VR44 are not yet easy to use certified, but all others.
VR44, it's easy to use. Sorry.
Okay, thank you. So it's a new.
VR44 bio is not.
Bio is not yet. Yeah, thank you, Etienne. Thank you. What happens? Okay, viability is okay. What happens with vitality of the yeast after in making different conditions? So if you see the next slide, we do 4 different trials for 4 different applications. One of them, when we added the yeast directly pitched in the mast, this you can see in green. DP means SH12, DP in green means direct pitch. The second one, R17, means we rehydrated the yeast in water in 17 degrees Celsius. And then R37 means that we rehydrated in quite special conditions, 37 Celsius. And the last one, 3717, means that we rehydrated the yeast on 37 degrees of Celsius, and then we make acclimatization from 37 to 17 as usual. And if you see, the medium was 230 grams per liter of sugar. Yan was 0.8% Yan's sugar ratio, which means that the Yan is around 180, 184 ppm of nitrogen. And the sulfur content was 50 ppm. Fermentation temperature was 15 Celsius. And now, if you see, we have three different graphs. First one, we did this this trial to see the fermentative performance in these different conditions: fermentative performance, fermentation kinetics on fresh yeast. Fresh yeast means that dry yeast rehydrated or directly pitched on the mast, and you can see that the fermentation curve is almost the same in each cases. That's perfect. That's nice. Now we go to the second graph. Here you can see a 2 years old yeast. Okay, 2 years old, made with forced aging. Forced aging means that we mimic the yeast. We treated the yeast in a special conditions that mimic a yeast which is 2 years old. In this second graph, you can see also there is no difference between the different rehydration or directly pitched versions. If you go to the third slide, where we have a 4 years old yeast, okay, it's also mimic. It's a 4 years old yeast. It also was forced aging, treated in a special conditions that mimic a 4 years old yeast. Here you can see a slight differences at the end of fermentation, and the difference is not big. So after at the end of fermentation, we have seen 3.5 days delay compared to the fresh ones, and also 1.5 days between usual rehydratation and direct pitch. So overall, we can say that our yeast are yeast that are certified, easy to use, certified, are very robust, and you can use, depends on your production, of course. Either you can use directly pitched on the mast, or you can rehydrate it in different conditions without losing the viability of the yeast, without losing the fermentation performance, especially maintain the fermentation performance. And finally, we don't have the graphs here, but the organoleptic profile of the yeast, of the wine at the end of fermentation and maturation. Whatever is the preparation mode, the organoleptic profile is equivalent with each other, it's the same. Thank you. Now I give back, after a small break, the voice to Etienne. Etienne, you can drink your coffee again and continue, because you became a little bit dry. Thank you.
Thanks, Yannis. So we saw the selection of the yeast first with the IFV, Institut Français de la Vigne et du Vin. We saw the evaluation of this yeast. Then we saw the production of this yeast under a dry form with a specific process to have robust dry yeast. Now, how we use, or what would be the guidelines to optimize thiol expression by the yeast. So it's not only you have a yeast that is able to release the thiols, it's also a way to say, how can I use this yeast to optimize this thiol expression? Because you will see that you can completely shut down the thiol expression in certain fermentation conditions. So here is very briefly some guidelines only on thiol expression and not on ester expression. So what we did, the microvinifications, still with the IFV on Sauvignon Blanc, and what we did actually was only, so the SH12, fermentation in 50 liters tanks we adjusted then the Yan sugar ratio to 1 with different nutrients. So we will study nutrients, and we will study different temperature, you will see. So what are the experimental parameters? As I said, two things. We applied different temperatures regime, so either 12-degree constant all along the fermentation, or 18-degree constant all along the fermentation, but we wanted to know, actually, to release and to optimize the thiols, if we start to do 3 days at 18 degrees C, so almost, let's say, all the growth phase of the yeast at 18 degrees C, and then go back to the room at 12 degrees C, so progressively decrease the temperature to 12 degrees C towards the end of the fermentation. And these three regimes of temperature were actually mixed and crossed with 3 different nutrition programs. So we adjusted the Yan at 1 ratio, Yan on sugar 1, but with different ways, and always with 2 additions. So 1 addition at yeast inoculation and 1 addition about 1/3 of the fermentation, so initial density minus 35. And what we did either first we put DAP, so diammonium phosphate, so ammonium source, and then spring ferm extreme being a total yeast totalizate that some of you already know, so it's completely organic. Or we do the reverse, organic first, mineral afterwards, or we do only organic at the beginning, organic at the at 1/3. And how were calculated the doses? It was calculated for the DAP, really function of its content in available nitrogen. For the spring ferm extreme, it was actually calculated taking into account that this total yeast totalizate has about 10% Yan equivalent. So meaning Yan equivalent is not the Yan, the real Yan, but it's what we think is equivalent to 10%. So if you see the results, that are the kinetics. The kinetics is very easy to see. Actually, you have here the densities, in let's say in dark, the densities, and in light color, you have the temperature. And you see for sure that this is the 12 degrees C constant. So start, let's say, and is delayed or slow fermentation, actually, when you are always at 12 degrees C. If you are at 18 degrees C, for sure, you start faster, you start faster and you go faster during the fermentation. So 18 degrees C, you finish at 18 days compared to 35 days in terms of 12 degrees C constant. And it's obvious, but it's really interesting to see that if you start at 18 and then you go progressively to 12, this is this. So you start and you almost mimic the curve of the 18. And then as far as you change the temperature, you start to slow down the yeast. And this actually shock to the yeast leads to have actually about the same delay, also the same duration of fermentation than if you were at 12 degrees C constant. So one of the main why we put this slide here is really for you to understand that temperature is crucial for the activity of the yeast. It's really the main important point to control when you're doing a fermentation. It's really drastically impacting the activity of the yeast. Then, so I don't go through all the parameters, but only on the thiols. So only on the thiols, I will explain this graph. So this is the thiol at the end of the fermentation. It's in OAV, so odor active value. So this is actually what you feel really. So it's the concentration divided by the perception threshold. And then you have the different conditions: 12 degrees C, 18 to 12, 18 degrees C. And in each temperature, you have DAP first, spring fermic stream, or spring fermic stream DAP, or only organic. And then you have described the three main thiols, as we saw, 3MH, 3MH acetate, and 4-MMP. What you see is that the SH12 has the ability to release 4-MMP. But what you see as well is that the 4-MMP is not present here. At 12 degrees C is not present. Meaning that if you want to release at least this kind of thiol, so 4-MMP, if you are too low temperature, you actually don't regulate the IRC7 gene activity. And if you don't regulate the IRC7 gene activity, you are not able to release the 4-MMP, even if you have a yeast that has the ability to release it. So you will only release 3MH and 3MH acetate. But what you see as well at 12 degrees C is that the acetate esters is higher than in 1812 or 18. And this is interesting because why? You will release 3MH, you will convert into 3MH acetate, but the acetate esters are really volatile, like isoamyl acetate, like other acetates are really volatile. So if you want to keep these kind of compounds, these kind of esters, for sure, if you ferment at low temperature, and especially at low temperature towards the end of the fermentation, you will keep a higher concentration of these acetate esters. And that's exactly the same way if you want to keep isoamyl acetate. So it's interesting to see that the acetate ester, maybe the conversion between 3MH and 3MH acetate is lower, but actually you keep higher concentration because of the temperature. Then what is really interesting, when you put the yeast in the conditions that it will be able to release 4-MMP, you can block actually this release when you use DAP at the beginning. If you see here, DAP at the beginning or DAP at the beginning, you can block or really lower the release of 4-MMP when you do ammonium at the beginning. And we will see why, because actually ammonium is delaying, as far as you have a lot of ammonium ions at the beginning, it will delay the uptake of the precursors. And if it delays the precursor of the uptake, it will also delay the release of the correspondent thiol. So it's a drastic difference. For example, in 1812 degrees C, drastic difference between DAP at beginning, spring fermic stream after, or spring fermic stream at the beginning that release a lot of 4-MMP. So we will try to explain a little bit this thing, just briefly. That's called the nitrogen catabolite repression specificity or pathway. What is the nitrogen or the Ncr? So the Ncr Regulation System. It's actually a regulation system within the yeast that makes the yeast taking preferentially the preferred source, the source of nitrogen that will allow its best growth. So it means that the yeast will take first the nitrogen source that are the best for its growth. So this will prevent, actually, it's a regulation system, the Ncr, that will prevent first the uptake of non-essential or non-preferential nitrogen source. And what we know is that actually the Ncr impact a lot on the opt1 and gap1, so this permease. It means that these precursors, also some amino acids that are taken up by this, let's say, permease, are non-preferential source. So as far as you have preferential source in front of you, in front of the yeast will preferentially take the better source and not the other source. So meaning that first, if you have a high nitrogen level, for sure this uptake of precursors will be shut down. But the same is that if you put too much ammonium, in the medium. This ammonium will also be preferentially taken up by the yeast, and so will also shut down the uptake of the precursors. And if you shut down the uptake of the precursors because these precursors are not essential for the yeast growth, it means that you prevent the uptake. If you don't have them, the precursors into the cell, you cannot release the volatile from these precursors. So it will prevent the precursors uptake, but we also know that actually this Ncr, so this regulation system, is also done regulating the IRC7 gene. So it will actually down-regulate the uptake, but also down-regulate the release. So meaning that if this Ncr is active, and when you put ammonium in a very high, let's say, concentration, you actually maintain this Ncr regulation system active, you will prevent the uptake and prevent the bioconversion. So especially for thiols, it's critical to have a good nutrition system if you want to optimize thiols. So as a conclusion and the key points, that's a lot of stuff in this slide, but mainly temperature influence is the stronger impact for sure. If you do at 18 degrees C, for sure you have a high release of thiols. If you do at 12 degrees C, you have a low thiol release or even no thiol release, but a high conservation of acetate. What is interesting is that if you do the compromise, so you start at 18 and you go to 12 degrees C or 12 degrees C in microvinification, you can do it. You can do this easily. But if you're in the winery, it will be 15 or 16. This is a good compromise because you will produce and release the thiols, but you will also maintain high concentration of higher alcohols and acetate esters. So you will have quite a high complexity, and it will be interesting. Regarding the nutrition influence, you have for sure, compared to the temperature, a moderate impact, but it's not that moderate if you take into account at least the 4 mmP, because if you take into account the 4 mmP, if you put DAP at the beginning, you really lower or even block the release of this thiol. So if you want to prefer acetate esters, you can put the DAP at the beginning. If you want to prefer actually thiol spring ferm extreme at the beginning or nothing, and again, spring ferm extreme and DAP at the 1/3 seems to be the best compromise because you have high thiol release, but also high esters maintenance. So we saw the selection, we saw the production of the dry yeast, how to use the yeast. Now it's time to go to the field. What we did last year extensively was field trials before commercialization of the SH12 was the trial in the field of the SH12. So it's always the same. You have the same. You ask the winery to have the same must divided into 2 tanks, exactly the same conditions in each tank, but one is a test tank with SH12 and the other is a control tank with the normal yeast the winery used. So we did 32 trials in 6 different countries on 15 varieties. So you see where we did the trials. And actually we did 1 trial in Greece as well. That was quite successful, was interesting. And the global overview is what? So it's the main parameters I will show you. Main parameters is, for example, implantation controls. For sure, when we do a trial, even if it's in a winery and is in the field, we need to know if our yeast made the job. And to be able to tell everybody and to exploit the results that our yeast made the job, we take a sample at the end of the fermentation. We analyze this sample in terms of Pcr analysis. And this Pcr will say, yes, in majority you have the SH12 or not. And if you have in majority the SH12, we can say that the SH12 made the job. So then we can interpret the aromatic profile or organoleptic profile. So what you see here is actually all in green is in majority of all the trials, the 36 trial or 32 trials, we had a high implantation rate, so SH12 was well implanted. Some of the kinetics, just to say that in different, so you see different trials. Overall short lag phase, always a short lag phase with the SH12 and the regular kinetic. So was completely acceptable compared to reference. Then if you go compared to the control, what was the malic acid consumption, the total SO2 production, or the others, actually what we saw is that you have to look into this scale. It's just, let's say, 0.6, for example, days less with the SH12, or 0.4 milligram per liter less SO2 production. So actually, all these parameters says that SH12 compared to the reference in the market is completely in the standards. And then that's our the tasting. I will not go through all the tasting, but what we saw is that we saw enhancing thiol-related notes, lowering the amylic notes always. It was lower in amylic than the control, with a nice acidity and a better balance. The same on rosé. We did only not only whites but also rosé. So now I leave the speech to Vicky that will explain what they did internally, the testimony on the SH12 trial at Innovino Lab.
Okay. Can you hear me?
Yeah, we can.
Okay. Thank you, Etienne and Yanis, for the very interesting informations. So I'm here to present you
the voice to Vicky, no?
Yeah,
Can you hear me, Yanis?
Yes, we hear you very well. It's okay.
Okay. So I'm here to present you my experience with SH12. In fact, here at Innovino, we feel very lucky for many reasons. And one reason is that we work with Fermentis people and Fermentis products. And another reason is that we don't only sell the products, but we can also evaluate them on our lab. So last year, when SH12 was not in the market, we had the opportunity to test its performance on a trial that we were organized with Fermentis. So here is the protocol of the trial. As you can see, we took grapes of Sauvignon Blanc from Ncr region, from Pyrgakis estate. We crushed the grapes, pressed the grapes, and then let the grapes to be settled, racked the wine, the must, added 4 grams per hectoliter sulfur dioxide, and the pitching rate of the yeast was 20 grams per hectoliter. I will present you the trial that we made with SH12 and 3 other commercial strains. The fermentation took place in 30-liter demi-jones in a cold room. And also you can see the initial analysis here. So the initial sugars was 210, and we had very good total acidity. So we didn't do any addition. Instead of adjustment of yarn, we wanted to be sure that we have an efficient nutrition, because we didn't want a lack of nutrition to affect our experiment. So we added yarn equal to sugars level. And the nutrition program was at the beginning of the fermentation and at 1/3 of the fermentation, as it is the common practice. So next slide again, okay. So here is our cold room. where we could take daily measurements and control of the fermentations. And also, we could also smell daily the fermented wines. And I can say that it was a big surprise for us, the performance of SH12, even from the first day from the first day of fermentation. Okay, let's move on. Okay, here is our lab where we do all the microbiological and chemical analysis. We evaluated many parameters, but for time saving, I will present you only the most important. So you can see here the sugar assimilation, the kinetic of the fermentation. And as you can see, SH12 complete the fermentation within 9 days. So we can say that SH12 has a regular to fast kinetic. Also, the other 3 yeast had a normal kinetic, so we didn't face any problem with stuck or sluggish fermentation to this experiment. Another crucial parameter, as Etienne already explained, is the Pcr analysis, because we want to be sure that the resulted wine is a result of the inoculated yeast and not from the indigenous yeast, or that there is no contamination between the tanks. So we applied Pcr analysis to 3 different stages of fermentation. And next slide. You can see here the results. So you can see the implantation of the yeast at 48 hours, at the 2/3 of fermentation, and at the final stage of the fermentation. And as you can see, SH12 is dominated from the very early stage of fermentation. You can see that at 48 hours, we have only 1 unknown colonies of another strain, and the 15 colonies are from SH12. Now, we know that SH12 has the killer factor, so it makes sense. We didn't know last year. And also, we can see that yeast number 3 has the same performance as SH12. And we already knew this commercial strain that it has the killer factor, so it makes sense as well. Yeast number 1, we can see that it is the strain that completed the fermentation, but it was not dominated from the beginning. And this strain is a sensitive strain. And about yeast number 2, we can see that it presents and starts the fermentations at the beginning, but it could not dominate. So the indigenous yeast took over and completed the fermentation. Okay, let's go on. Another crucial parameter is the malic acid concentration. As you may know, all yeast strains degrades the malic acid, and the percentage of malic acid degradation of SH12, it was very low, as you can see also on this diagram. This is very important to white and rosé vinifications, especially in Greece, where malic acid is too low. Most of the time it's less than 2 grams per liter, and we want to preserve the natural acidity and freshness of the wine. So at the next slide, you can see that total acidity was correlated to the malic acid concentration. So we can measure, we can see higher total acidity for SH12. Next, please. Okay, and the most important part always is the tasting. So last February, we presented the wines and the results at an exhibition that was organized by the Union of Artisanal Wineries of Greece. So we had 80 participants, winemakers, enologists, and sommelier that tasted and evaluated the wines. And after the statistical analysis, you can see on the spider diagram the results. So you can see that the wines from SH12 with the yellow line were evaluated as more acidic. And here more acidic, yes. Much more tropical fruity. Here, right? Less estery, more citrus, higher intensity of terms of aroma, a good body, better balance, better aftertaste, and after all, a higher score at the overall quality level. And I will stay at the tropical fruits aromas, as afterwards we conducted thiols analysis on our samples. And as you can see from the PCA statistical analysis, SH12, it's clearly thiolic, as it is correlated to the thiols. So, the conclusions that we made is that SH12 is a great choice for thiolic varieties. It has a very good implantation. It has a normal kinetic. It has low degradation of malic acid. And for us, this is very important as it gives more crispiness and freshness to the wines. It has very high production of thiols, especially for MMP. In some cases, it was 2 to 5 times more than the other yeast. And of course, the most important, that it has a high score at tastings. That's all. Thank you. Etienne?
Thanks a lot, Vicky, for this testimony. It's very interesting for us as well, the partnership with Innovino for several years now. Always a pleasure. So we saw, I think you can maybe mute your computer.
Yes.
We have echo, yeah, perfect. So I will just finish on this very quickly, because I see we are running a bit late. So what are the conclusions? So on this SH12 characteristics, it's very easy. Technical characteristics, very good implantation, good alcohol tolerance, wide optimum temperature, low YAN requirements, but mainly what was interesting, so high total acidity, low production of S2, H2S, high thiol release and conversion. And for sure, it's killer and easy to use. So on the technical characteristics, it's quite a clean fermenter, regular fast, with a high thiol, let's say, capacity, high thiol release capacity. So in terms of targeted wines, it's very easy now. It's really targeted. Fruity, white, and rosé wines, rich and complex in thiols. Why rich in thiols? Because of the release. Why complex? Because you have all the thiols, not only the fruity ones, but also the vegetal green one, and also the complexity coming back from the ethyl ester production. So what we saw actually on the ethyl ester production is that on specific white varieties, it was enhancing the white fruit notes along with the thiols. In the rosé, it was especially on the red fruit notes or small, let's say, red small fruit notes, but not masked by amylic notes anyway. So it was interesting to see that. Very easy to drink, a nice balance, crisp and fruity wines. So I don't know if you're familiar with this aromatic mapping, but actually I will just briefly say that the SH12 among all our white and rosé yeast will be placed here. So promotion of the grapes aromas, so on the thiols, we saw it. and will be on the low axis, so on the low sweetness roundness, because high crispiness, high, let's say, acidity remaining. In terms of technical mapping, as we saw, it's not a yeast that is really fast from the beginning to the end, but it's fast at the beginning and then acceptable to the end. So it's on the regular fast, actually, kinetics, and so resistance, low nitrogen needs, and it's in blue because actually you saw it already, some experiments at 12 degrees C, for example, and it was okay at 12 degrees C and was actually completely comparable to fast yeast in that case. So low temperature tolerant. So that's it. I think we got a global overview of the SH12 characteristics and evaluation. I didn't see actually on the chat a lot of questions, or even no, I think, questions, but maybe I didn't see everything. Oh, yeah. So now maybe I ask Gilles to come back on board, and maybe if we have some of the, let's say, some of the questions.
Yeah, actually, yeah, correct. There weren't only one questions from Delcio. So he asked if we have a protocol. So I'll let you answer that.
So I think it's a protocol to use the SH12, yeah, and to release more thiols. So it will be done. So in let's say with Yanitz and with our distributor. So to put in place a protocol, if you don't need a lot of nitrogen to be added in your let's say in your mast, in that case we will only advise to put nitrogen and organic nitrogen at 1/3 of the fermentation, and nothing at the beginning. Nothing at the beginning, to let the yeast be able to, let's say, release the thiols or uptake the thiols and then release them. If you have a big deficiency, as we saw, we will advise organic first and then DIP at 1/3. And for sure, in the temperature, if you want to favor the thiols, we will never tell you to start at low temperature or very low temperature. Always start at, let's say, 18 degrees C or a little bit more, 18, 20 degrees C, for the yeast to release the thiol as much as possible at the beginning. So yes, the protocol, we can build one with our distributors and our sales managers to be transferred. Yeah, for sure. So that's one of the questions. Maybe we have some others or not. I don't think so.
Yeah, at this time we don't have any more question from the audience, but if you do have questions,
yeah, don't hesitate, actually.
Or I think there is even a tool to raise your hand. And if you do that, I can give you access to your microphone in order for you to ask the question live, if you prefer, of course.
I see Dimitra is typing something, or maybe not. Or maybe Vicky, you have some questions as well, depending. Oh, what about skin contact? Is it preferable to do it or not? So this is something that is always interesting on thiol. Actually, most of the time, this skin contact is preferable. But I could give you, let's say, a paper, let's say, talking about that. I'm not this specialist, but for sure, if you allow skin contact, you allow the precursor to go into the mast, and so for the yeast to be able to take them. So I would say actually it will work. Skin contact will actually enhance the thiol release, as far as the skin contact will be in a controlled atmosphere. So would be done, for example, in in an inert press or these kind of things, because these compounds are quite actually sensitive to the oxygen. So as far as you do this skin contact, but in controlled atmosphere, it would be preferable. But I can, there are some papers on that. I'm not specialist about that, and we could transfer if you want. How does it work on fruits? So it depends on the types of fruits. Actually, I will say, I don't know if a lot of fruits has thiol, let's say, precursors. I know in apples there are some thiol precursors and some terpenol precursors as well, so it should work quite well. And apple is actually quite close, not completely, but quite close to grapes. For the others, we need to know a bit more the parameters. And Stelios, initial NTU with SH12 to promote more the thiols. Yeah, in most of the case, the initial NTU, I always advise if you can more than 50 NTU, because if you go, let's say, below 50, you start to have some other problems in the fermentation. And anyway, if you release more thiols, they will be lost somewhere because you will produce off flavors. So I would prefer, say, 50. The best would be around 100 NTU. It will not affect your wine, and it will for sure help the fermentation to go better. So help as well the yeast activity on the thiol. Yep. So Georgia is saying, no. It was a wonderful presentation. Oh, thank you, Georgia. This is good news for us. Happy to work with you as well. Okay. Oh, Ruth, you are here? That's nice as well. We have also colleagues from New Zealand actually with us, so you have to know that. New Zealand, let's say the country of the Sauvignon Blanc. Huh? I think with South Africa, with Chile, France, these countries are really let's say almost interesting by the thiorides and thiovarity because of the Sauvignon Blanc typicity.
Okay, we have a question from Sanjay. Is it suitable for
citrus fruit?
Yeah.
Actually, as I told you, the main problem of citrus fruits is the pH. So the pH and the type of pH type of sugar you have, but most of the time in the fruits, except let's say in the grapes, it's mainly fructose. So we didn't see any problematic of this yeast to use fructose, but for sure it's not among the best one to use fructose. That's why you have at the end of the fermentation, let's say a slowing down of the fermentation, but acceptable. So I would say it's fructophilic, but not that much. So for citrus fruit like orange or others, I think the pH will be the main problem. So as far as you can regulate the pH, we could try, yeah, or use other yeast that are above this yeast in the chart in the map, like BCS 103 or CKS 102. These kind of yeast are more fructophilic, let's say, and will be maybe more suitable for citrus fruit. Yeah. But anyway, we don't have any data for time being with the SH12 on the citrus, so it could be a trial.
So we have another question from Anthony. Have you quantified the percentage of 4-MMP during the alcoholic fermentation? So I guess that could be to monitor when the 4-MMP appears and the way it evolves during the alcoholic fermentation.
So what we know from the literature is that these kind of 4-MMP, 3-MH are rather released early in the fermentation, so during the growth phase of the yeast, because actually during the growth phase you have the energy, you also have the activity and the production of more catalyzer. And in that case, the 4-MMP, 3-MH appears mainly at the beginning of the fermentation, but we didn't quantify it during our microvinification. We always took the final point. So we didn't let's say study the precursors and the conversion and actually the kinetic of conversion of these precursors during fermentation. For us, it's sure that these kind of questions is really interesting, but it's much more a research question, actually. And most of the time, yes, what we need at the end is the concentration at the end. But it's a good question. We didn't do that. Ruth, Larry, yes, I am.
So our colleague Ruth is very interested to get the yeast. So that's a good thing, I guess. Hi, Ruth.
Just for everybody to know, Ruth is our sales manager in New Zealand, Australia, Japan, Korea, and we have several microvinifications going on in New Zealand on Sauvignon with the Research Institute, but also in South Africa with the Stellenbosch University, to assess this yeast on Sauvignon Blanc for these very typical countries.
Okay, Stelios is asking, I think, an interesting question. Do we have experience with less tyric varieties like Chardonnay? And then he's talking about the same philosophy of 18 to 12 degree temperature management. What I would guess from that is that it would be the behavior towards ethyl esters when the potential of thiols in the grape is not as high as Sauvignon Blanc, for example.
Yeah, I actually didn't say anything about the ethyl esters and acetate esters in the presentation, but actually I have some slides that were hidden actually. So let me see if I can go to one of them just to let you know what we had. I know that Inovino did the trial on a non-thiolic variety, so they have some data on it. But for example, yeah, so it's a bit maybe difficult to see.
I confirm it's pretty small on the screen.
But that's the same. That's the same way as I did for the thiols. You have the 12 degrees C, 18 to 12, 18, 12. So okay, and then the three conditions: DAP, spring firm, or spring firm DAP, or spring firm, spring firm. Overall, what I tell for the 3-MH acetate is the same as isoamyl acetate. So if you ferment at very low temperature, this yeast will produce quite a lot of isoamyl acetate and maintain it. As far as you go towards the 18, for sure, you produce less isoamyl acetate. So that's for sure. If you want to produce more, we did that actually for with for example, the CKS 102. One of our yeast that is actually also used on the thiols, but releasing a lot of acetate esters. And I ensure you that if you ferment at 12 degrees C constant with the CKS 102, whatever the variety, you will have a pure banana juice. So it could be interesting, depending on if you want a pure banana juice in a tank and be able to, let's say, mix with other to do a right blend. But for sure, if you want to produce more acetate esters, you need to ferment at lower temperature. For ethyl esters, it's a bit the contrary, but it's not actually big differences between temperatures. It will be a little bit more let's say, ethyl esters also at high temperature. Why? Because some of the esters, like octanoate and decanoate, ethyl decanoate and ethyl octanoate, are long chains, and these long chains are more able to diffuse through the membrane when you are at higher temperature, meaning that you will produce, or at least having more concentration of this kind of ethyl esters when you ferment at higher temperature. So if you can say you will decrease the acetate, you will increase the ethyl esters. So if you want to balance ethyl acetate, and in each variety, let's say, because these are fermentation derived aromas, it will be this case. For 2-phenylethanol and its acetate, it's a bit different, because 2-phenylethanol and acetate, you first need to produce the 2-phenylethanol and then to convert it to acetate. So for sure, the higher the temperature, the higher concentration of 2-phenylethanol, because higher alcohols are more produced at high temperature. And because of that, this acetate esters is actually also related to the high temperature. That's why 1812, it's interesting if you want a mix of everything. You will have quite a lot of isoamyl acetate, quite a lot of ethyl esters, and quite a lot of floral notes, so phenylethanol acetate. And this could be applied actually mainly to most of the variety, if you don't have any thiols or terpenols that are varietal. So I don't know, maybe we can ask Vicky about her trial on another variety in so non-thiolic variety in Greece. I know if Vicky is there. Yeah. So just to have an idea about this, so I don't remember on what you tried.
We didn't do the analysis of the aromatic compounds. We did only the tasting. It was interesting, but the data of the aromatic compounds.
Okay. So I don't know if it's answered your question, but really starting at 18, so starting high temperature and then go to a lower temperature, most of the time it's interesting when you have varietal aromas. But what we saw is that it's also interesting in terms of ester production. So here, and here. Okay. So I don't know if we so Vicky, maybe if you can just mute your microphone. Sorry, but we always have an echo.
Yeah, apparently Stelios said thank you. So I guess you answered his question the way you wanted.
Okay.
Good thing.
We have a question of Yanis. Please add me on moderators.
Yeah, I'm looking for him. Yeah. Here's Yanis again.
Okay. I think we exceeded, let's say, of about 15 minutes the webinar. I still see quite a lot of participants, but I think we can maybe close the session, Yanis, if you agree with that.
Yeah, I agree with that. And please contact, I ask all participants, if you have any further questions, I ask you to send the questions to your distributors, or on Fermentis page, you can have the opportunity to send your questions related to this webinar, and we will answer for you for sure. So this is please add your emails, addresses, etc. I saw on the list of participants that all the distributors, almost all the distributors for Central Europe and also from Caucasus region, Georgia, Armenia, Romania, also from Central Europe, and also Hungary, Poland are here. Thanks for that. So please send your further questions, and I really thanks for all participants participated on this event. That was our first wine webinar event in Central Europe under the umbrella of Fermentis Academy. For your information, Fermentis Academy is our forum where we can share and exchange our informations and learn the knowledge of each other. So we think that we will continue this soon. Maybe not initiated from Greece, but from Hungary or from Poland. So we have the plan. So we continue. Thanks for all the participations and say hello for everybody. And thank you again, Etienne, Vicky, and Gilles for your really valuable participation, and also for Ruth from New Zealand. Her participation as well. Thank you again. Have a nice day. And this event and also the whole presentation is recorded. So hopefully we can share this presentation also with you, so you can study it again and again to be more stable. The knowledge remains and remains with you. So thank you again. Have a nice day. Goodbye.
Yeah. Bye, everybody. Thanks, Vicky.
Bye.
Thanks, Gilles. Thanks, Yanit. And thank you all. I will stop this webinar. And yes, and great harvest to all.
Yeah, for sure.
Bye-bye.